There is no more important fundamental concept in human physiology than molecular transport. It underlies and drives almost every physiological process, from how your brain communicates with other parts of your body, to how your heart beats, to how your muscles contract, to how your kidneys filter. All of that is driven by molecular transport. Now, what I mean by molecular transport is, imagine if I have some solute or water out here and I want to get it in the cell. That's molecular transport. Or if I have something in the cell and I want to get it out of the cell, that's molecular transport. Notice that in both cases, I need to cross the plasma membrane. We're going to start our discussion by building on concepts learned in Chapter 11 of the workbook when we learned about diffusion. So diffusion is a natural process that occurs because molecules are constantly moving. So if I take a bunch of molecules, each one of these is constantly moving around. And so the faster those molecules move, the faster I'm going to see diffusion occur. So it turns out that there are two things that impact how quickly molecules will move. One of which is the temperature. The warmer the temperature, the faster they move. So in a refrigerator, a molecule might move like this slow, really slow, where at 60 degrees Celsius, it might be moving like this, really, really fast. So the faster molecules move, the faster they diffuse. So temperature can increase the diffusion rate. Another thing that affects how quickly molecules move is their size, or molecular weight. A small molecule will naturally move faster than a big, heavy molecule. So if the temperature is the same, and I have two molecules, a small one and a big one, the small one might move this fast, and the big one is going to lumber around, kind of move really slowly relative to the small one. So the smaller the molecule, the faster it moves, and therefore the faster we reach diffusion equilibrium. Okay, so now let's look at another feature that can affect diffusion rate, and that is distance. This one is a little harder to explain, but I'll do my best. Imagine that I have two compartments, a really long one and a short one. I'm going to go ahead and put the same concentration of dots on the left-hand side of each compartment. Which line do you think they're going to cross first, A or B? If you said B, you're correct. Again, just random motion. They're going to start crossing the dashed line that represents B faster than they're going to cross the line for A. So the farther the distance, the slower the diffusion rate. So if I have a compartment here, and we'll just call this X, and a compartment here that we call Y, it's going to take a long time for those molecules to get from X to Y. If I want to get them to Y faster, I need to bring X and Y closer together. In this course, we are going to see some real applications of this, especially when we get into the lungs and we look at gas exchange between the lung airspace and the blood. The final factor that can affect diffusion rate is what is called the strength of the concentration gradient. Now, I really hate that phrase, but it is one that is used, and so we'll use it too. The strength of the concentration gradient is a way that we measure or describe how concentrated something is. The red dots have a stronger concentration gradient in the B box rather than the A box, and that is because they are more concentrated in B. So A represents a weaker concentration gradient, and B represents a stronger concentration gradient. Again, not my favorite descriptive term because strength usually represents, like, you think muscle strength, the amount of weight that you can lift. But here it's referring to how concentrated those particular molecules are. So the stronger the concentration gradient, the faster the diffusion rate. Let me go ahead and write that out, and then I'll describe it for you. There are a few different ways to measure diffusion rate. In our case, I have a dashed line across the diagonal of the box. What I want to know is how long is it going to take until, let's just say, 3 molecules cross that dashed line. And in my A box, these guys might be moving around a little bit like so, and it might take them a while before I eventually get 3 across. But in my B box, they're all so crowded that they're actually naturally going to want to spread apart. Even before COVID, everybody likes their personal space. And I want you to imagine being in a really crowded street and where you're basically packed in like sardines with other people, and you see an open spot. You gravitate towards that open spot because your personal space feels, impacted. And in the same way, molecules bounce off each other and ricochet like this, and then kind of move quickly across that line. The stronger the concentration gradient, the more the molecules are going to ricochet off each other and away from each other, and therefore the faster the diffusion rate. So those are 4 factors that can affect the diffusion rate. In terms of physiology, our body temperature should be kept constant at 37 degrees. We don't see a huge deviation in temperature while we're living, and therefore we don't see a huge deviation in diffusion rate. Moreover, molecular size or molecular weight is intrinsic in that molecule. It's not something that can be changed. So something that is tiny, there's nothing more tiny than a hydrogen ion, will move faster than a big lumbering protein. The protein and the hydrogen ion aren't changing their size. However, there are diseases, for instance, that can increase the diffusion distance and therefore reduce the diffusion rate. Likewise, if we want to get molecules to diffuse quickly, we can increase the concentration gradient to make that happen. So those are 2 factors where there will be real physiological consequences, and when they don't work properly, that can be a state of disease. So now that we've looked at diffusion, we're going to turn and take a look at the plasma membrane. Remember that molecular transport is almost exclusively concerned with crossing that plasma membrane. And if I am some substance, and again, I'm going to use my generic X there are really 2 choices. One of which is to try to get through the phospholipid bilayer. And if you can do that, you're basically playing a game of Frogger. You're waiting for these phospholipids to move apart, and then you're trying to kind of slip through the cracks. In order to be able to do that, you have to be small and not too terrified of the center. And so there's really only a handful of molecules that can do that. And they are carbon dioxide, oxygen, and then water makes it through as well. And water is a really interesting one because we tend to think of this core of lipids being hydrophobic, which by definition means to repel water. So when I tell you that water can slip through occasionally, it's kind of surprising. But it has to do with the fact that there is just so much water that there's enough gaps that open up in that membrane. The membrane is not static, that water can slip through. And I'll give you another analogy here for how water can slip through, at least some of the time. In San Diego, we have the I-15. And the I-15, I think, is about like 5 lanes each way. Let's go ahead and add some lanes here. I'm ignoring the carpool lane. And of course, we have cars. And those cars are moving really fast. Now I want you to imagine that I have a bunny. This is not drawn to size here. But let's say that I have a little bunny. And the bunny is going to try and make it across without getting killed. So it's got to make it across all these lanes to the other side. That's like a water molecule trying to make it through the phospholipid bilayer. And I ask you, what are the bunny's chances? And you go, pretty small, especially if there's a lot of cars on the road. The chance that it's going to get squashed is pretty high. And that's true. The chance of a single bunny making it across is very low. But what if I have 1 million bunnies and they all try to make it across? Then one might get through. So, in other words, one bunny by itself probably isn't going to make it through, but if I have 1 million bunnies, a few of them will. And water is the same way. The most common molecule out here is water. And in here, water. And therefore, some of them are able to sneak across. Okay. Occasionally, a lipid can sneak across, but it's not common. So steroids, in particular, are kind of the other one that are like, in small quantities, steroids can slip across as well. Everything else is going to require the help of a protein transporter. So they're going to need to be able to get through some sort of membrane protein. And so on that note, let's actually take a look at the various proteins that are in a plasma membrane. You know, up to 50% of the weight of your plasma membrane is protein. So there's a lot of proteins and they do a lot of different things. The first thing to note is that not all proteins are going to span the membrane. Some will span the membrane, and we know those as integral or transmembrane proteins. And then some sit kind of off to the side, and we know those as peripheral proteins. But in physiology, we're less concerned with anatomy and more concerned with function. So let's take a look at some of the functions of these proteins. There are 4 major classes of membrane proteins. One of which, and perhaps the most important to us, is transport. So again, these are proteins that are going to be embedded in the plasma membrane. Another of which is to be a receptor. Some membrane proteins are enzymes, and some are used for structure. So here I have an example. In my class, I get very tired of drawing out all of the phospholipids, so I tend to use a line like this to represent the plasma membrane. And you can see I've got several membrane proteins embedded in that plasma membrane. This right here is a channel protein. So a channel protein is going to allow some small thing safe passageway across the plasma membrane. So something other than CO2, oxygen, and maybe a steroid. The next picture over is both an enzyme and a receptor. So receptors very often have their face facing the extracellular fluid. So in fact, let's go ahead and write that in. So if this is a plasma membrane, and this is just my designation, I almost always put the extracellular fluid on top and the intracellular fluid on the bottom. A receptor is going to bind to some signaling molecule. So here I have it as a green, like maybe protein, and it will bind like so. And this particular receptor has an enzyme on the other side. And so when that receptor binds, it almost acts like an allosteric activator. And I'm going to go ahead and change the shape of this enzyme now. And it's going to open that enzyme up for business. And so now this enzyme can run a chemical reaction, can catalyze a chemical reaction. So I've actually made a membrane protein, a transmembrane protein, that is both receptor and enzyme, demonstrating that you can have proteins that do multiple things. This last one over here, this is a structural protein. It's anchored into the plasma membrane, and then it's hanging off and basically binding the cytoskeleton. So that's a description of the 4 different types of membrane proteins. Now, I want to come back to the receptor. Receptors are going to be important in some of our physiological processes. And so we have a name for a molecule that can bind to a receptor. It is called a ligand. Since we're focused on molecular transport, we are going to take a closer look at these. Membrane proteins that are involved in transport usually come in 2 different varieties: channel proteins and carrier proteins. You were introduced to both of these in Chapter 11. Channel proteins will always be drawn like this. So that's my typical channel protein when I draw it quickly. Carrier proteins are going to be drawn maybe more like this. So channel proteins allow for diffusion of some substance. And if we take a closer look, we realize that channel proteins themselves actually come in a couple different forms. So this picture right here shows a channel protein. This is the plasma membrane, and you can see they kind of draw it just like I do. Extracellular fluid always on the top, intracellular fluid on the bottom, and it allows for movement both ways. It's going to be whatever the gradient is. So a channel protein will facilitate diffusion of some substance. But channel proteins can be open all the time, or they can actually have kind of like a flap on them. So here it looks like a ball and chain, like old school jail. I almost think of these like the lid of a garbage can, and the lid can open and close respectively. Something like that and that. Open, closed. So when a gated channel is open, diffusion is allowed to happen. When a gated channel is closed, diffusion stops. Channel proteins are built one way or the other. They are either built as open channels, and sometimes we call these leak channels, or they are built as gated channels. You can't decide one day I'm going to be an open channel and tomorrow I'm going to be a gated channel. You either come one way or another. The advantage of a gated channel is that a gated channel can be closed, and therefore you can stop the flow of whatever that particular substance is. So let's go ahead and add this to our list. I draw my gated channels with the gate kind of in the center. Okay, something else about channel proteins that's really important to note. The hole that channel proteins have is actually much smaller than what this looks like. Here's a more accurate picture of a channel protein, and you can see it is built of multiple cylindrical proteins. So here's a protein, here's a protein, here's a protein, and then the hole is actually quite small. So channel proteins have pores that are very small, and there's a reason for that. So an analogy that I like to use is a doorway. Imagine that you have an open doorway that allows whatever it wants to diffuse through. And if you have a room, and I'm just going to put a doorway right there, and there's a bunch of people in this room, and they're all really crowded, then naturally they're going to want their space, and if they see an open doorway, they might go to a less crowded area, like maybe outside. You know, you can think of a party that got too crowded and people start trying to move outside because there's more space. But that doorway is also going to be big enough to allow for flies, mice, dogs, etc. It's not going to be specific to humans because it has to be big enough for humans. So therefore, it's not real specific. In other words, yeah, sure, it allows humans. It also allows mice and rats and dogs and cats and whatever else to get through. And in the same way, if a channel protein got too big, had too big of a pore, then too many things would be able to get through. And it would no longer be selective. It would no longer be able to pick and choose what it allowed safe passage. And we do want our channel proteins to be specific for certain solutes. So, for instance, let us say that I have a channel protein that is specific for the red dot. And the red dot is going to be sodium, a sodium ion. Now, that channel protein, as I've drawn it, has a big enough pore that it's going to allow sodium to diffuse through, but not so big that it's going to allow a glucose molecule through. So glucose is prohibited. What if I tried to instead build a channel protein explicitly for glucose? Let's see what that would look like. In order to do that, I would need to make a bigger pore. Great, now it's big enough that glucose can get through. But you know what? So can sodium, because sodium is smaller than glucose. So by making my channel protein big enough to allow glucose to diffuse, I also let sodium diffuse, and anything else that's smaller than glucose, which is a whole lot of things, water, all kinds of stuff. I don't want that. So channel proteins are only used for the really small stuff, ions and water, because if we start making our channel proteins big enough to accommodate larger molecules like glucose, it no longer is selective. It's like an open doorway for a human that also lets the flies in, the cats out, the dogs in, etc. And we can't have that. So if we want to allow glucose to move, we're going to need a carrier protein. All right, and by the way, when we talk about channel proteins, we usually name them by what they allow across. So you've already encountered aquaporins. Those are going to be channel proteins specifically for water. But if I have a channel protein that's specifically for sodium, I'll call it a sodium channel. If it's specific for potassium, I'll call it a potassium channel. All right, now let's take a look at our carrier proteins. Carrier proteins can be useful for both diffusion and creating a concentration gradient. Now you saw an example of a carrier protein creating a concentration gradient with the omnipresent sodium-potassium pump. That sodium-potassium pump is so important to physiology that you might as well know it now because you're going to see it over and over and over and over again. So just so that you remember, the sodium-potassium pump is going to spend one ATP molecule and it kicks out 3 sodiums and brings in 2 potassiums into the cell. A cute way to remember that is the cell says nah to sodium, but K is a keeper. So that's how I always remembered that sodium was excluded, because I'm the cell, and the cell says nah to sodium, and K is a keeper. So the sodium-potassium pump is a carrier protein. And as a carrier protein, it is going to always be closed at one end. So hopefully you guys saw the video on this. This is just an image showing that. And what I wanted to show you is that notice that there's always one part that's closed. It's never open all the way through. And so it literally gently carries whatever it is across that plasma membrane. If you carry one thing across, you are a uniport, uni meaning one. If you carry 2 things across, like the sodium-potassium pump does, then you are called a co-transporter. And you can see that here. Some carrier proteins carry only one thing across. Some carrier proteins carry 2 things across. Now you can carry them the same direction, or you can carry them opposite directions. I don't care for those terms, symport and antiport, so I don't ever use them. Just know that there are certain carrier proteins that carry one thing across, and certain carrier proteins that can carry 2 things across. So let's add that into our table, and then we're going to be done with this part of the lecture. Okay, so something I forgot to write up earlier, but definitely want to make sure that you understand, is that if you ever want to create a concentration gradient, you have to use a carrier protein. It doesn't matter what size you are. Okay, that's where we're going to stop on this one, and I'll see you for the next lecture where we compare active and passive transport.